Hello and welcome to the 7th Annual Global Summit on Kidney Disease Innovation, titled The Decade of the Kidney at Midpoint: Opportunities and Barriers to Greater Innovation and Freedom from Disease and Disability. This annual event is hosted by American Association of Kidney Patients in collaboration with our allied partners at George Washington University School of Medicine, Sciences, and Health. My name is Moses Osino, and I'm honored to serve as AAKP International Ambassador from Kenya. I was diagnosed with end-stage kidney disease in 2017 and have been on dialysis since then. I dedicate to lend my voice to educate communities, and I founded the FIGO Initiative, a non-profit organization that creates awareness through educational forum and kidney health talk. Hemodialysis in Kenya comes with a lot of challenges since the government only pays for 2 sessions a week, leaving patients to dig deep into their pockets. Before I became critically ill, I used to do business, but I had to dispose few of my assets. Being involved in AAKP allows me to realize my dream mission to connect globally as kidney health matters are close to my heart. Since 2019, the Global Summit has served the global kidney community as the largest patient-led kidney disease innovation summit in the world. This year's event marks a critical milestone because it is important. It is the midpoint of the AAKP-led initiative, the Decade of Kidney. This global effort has been mirrored across the world by kidney advocates, medical allies, government officials, and among people of goodwill committed to a return of true patient-centered medicine. Throughout the first half of the decade of the kidney, we have raised awareness about the increasing burden of kidney disease worldwide among people, communities, workforce, and government. This effort has also accelerated the determination to transcend status quo treatment, including brick and mortar dialysis, to a future of kidney care that will be determined by expansion in patient consumer care choice, targeted therapies that stop or slow the progression of kidney disease and prevent kidney failure. Greater access to donated organs, new devices, and alternative to human organs, including artificial, wearable, and implantable kidneys and xenotransplantation. We believe the time is now as we move through the next 5 years of this golden age of kidney medicine, where much progress has already been made. Patients, caregivers, clinicians, researchers, innovators, industry, and government leaders to accelerate our bold action to push forward modern diagnosis, devices, and therapeutics, and to continue to remove outdated policies that block adoption. No country is immune to, impact of kidney disease, but the good news is every person has a role in shaping the future for all people who are at risk for or living with kidney disease that can offer more hope, better outcome, and greater independence and freedom from disease and disability. With that, I am honored to serve as the moderator for our titled Current Research on APOL1 NIH Leadership in Rare and Genetic Kidney Disease. We are pleased to have Dr. Jeffrey Kopp, Chief of the Kidney Disease Section, Kidney Disease Branch at the National Institute of Diabetes and Digestive and Kidney Disease within U.S. National Institute of Health, join us today. Dr. Kopp is a distinguished researcher in the field of nephrology, leading a translational research group studying focal segmental glomerulosclerosis, FSGS, and related podotide disease. Throughout his career, Dr. Kopp has focused on illuminating the factors contributing to the disproportionate prevalence of those diseases among African Americans. His groundbreaking work led to the identification of APOL1 genetic variant as the primary driver of his health disparity. His focus has now shifted to understanding how this variant influences cell function, with the aim of developing targeted therapies. Dr. Kopp also served as the consulting nephrologist at Mark Hartfield Clinical Research Center and as adjunct professor of medicine at Uniform Service University of Health Sciences. Please welcome Dr. Jeffrey Kopp.
Thank you to AAKP and the George Washington University for having me here today to talk about APOL1. This is an opportune time to have that discussion. This is the decade of the kidney at Midpoint, so 2020 to 2030. And we have opportunities and also barriers to greater innovation to bring our patients into freedom from disease and disability. The particular topic I'll be talking about today is an important genetic variant that drives FSGS. I'll give some background about focal segmental glomerulosclerosis and then spend a lot of time about this genetic variance in the gene apolipoprotein L1. So I'm talking to you from Pocatello, Idaho. I've retired from NIH. I live on this apple lane that you can see at the bottom. And I spent 37 years at NIH, got excellent training, and ended up focusing on FSGS and particularly on the role of APOL1. So this is very basic background. I thought I'd begin by saying we all have two kidneys, right and left, unless we've lost one due to trauma. Each weighs about 10 to 12 ounces. Here you can see one kidney on the right that's been bisected. You can see the red artery bringing blood in, the blue vein taking blood out, and then the ureters are the discharging the urine down, ultimately reaching the bladder. So what are the kidney functions? Well, most prominently is to remove waste products from the blood. Two that we measure frequently in clinical medicine are urea, an end product of protein breakdown, and creatinine, breakdown of muscle, both of which are convenient measures to determine if the kidney is filtering well. And if not, those substances, urea and creatinine, rise in the blood. Another important kidney function is to produce the hormone erythropoietin, which is essential to drive red blood cell production in the bone marrow. And if somebody has progressive kidney disease that scars the kidney, then the cells in kidney that produce EPO or erythropoietin are not functional. And so somebody has to receive what's available now, which is injections of erythropoietin. Another kidney function is to activate vitamin D by putting hydroxyl groups on it to make 1,25-dihydroxyvitamin D. And this is the active form of vitamin D. If you take plain vitamin D, which you'd buy at the drugstore, your body adds particularly the 1-hydroxy element. And this promotes calcium absorption from gut and bone. People whose kidneys are not functioning do not make sufficient 1,25-dihydroxyvitamin D, but fortunately can receive it supplementally as injections or take larger doses of plain vitamin D and hope that the remaining kidney function can produce that. And this explains why patients with chronic kidney disease need to attend to their bone status. In the days before we knew this, say 20 or 30 years ago, patients got significant bone disease, which fortunately is much less of a problem now. So ESKD, end-stage kidney disease, has a very high incidence in our country. About 100,000 patients per year have lost significant kidney function, nearly complete kidney function, down to 10% of normal, and require either a kidney transplant, which is the preferred therapy, or to go on dialysis, of which there are two forms. There's hemodialysis, where you receive treatments 3 times a week, typically in a center, but it can be done at home, or peritoneal dialysis, where fluid is put into the belly 4 times a day, and the toxins are removed that way. So at the top left, you can see the rates of kidney disease have been rising over the last 25 years. From 1980 to 2004. That's unfortunately, I don't have a more updated slide, but I can tell you it has continued to increase, particularly for most of our populations, with African Americans having the highest rates. And I'll be coming to discuss why African Americans are particularly prone to chronic kidney disease. So at the bottom left, you can see the lifetime risk for ESKD, end-stage kidney disease that requires therapy, is about 2% in whites and about 8% in blacks. And I'll be spending time on the genetic variants that drive that difference. Now, in terms of causes of chronic kidney disease, turning out to the right side of the slide, diabetes leads the list, and the African American to European incidence ratio is about 3.7, so more kidney disease in African Americans. For hypertension attributed kidney disease, the African American ratio is even higher, and I'll be discussing why that is. And I say it's attributed because we now understand that there's a gene that I'll be talking about, APOL1, in which variants drive kidney disease. So some people are saying hypertension misattributed. It's really the kidney disease driven by APOL1 variants that then leads to hypertension. But this is still some of the old terminology. Glomerular disease, 2.7 increased rate in African Americans, driven by both focal segmental glomerulosclerosis, which I'll be talking about in more detail, and AIDS nephropathy. Now, a more recent term would be HIV-associated kidney disease. Fortunately, with effective therapy for HIV, this is a disease that is rapidly declining, possibly even disappearing. So overview of glomerular diseases. Again, the glomeruli are the filtering units of the kidney, and that's where most kidney disease starts rather than in the tubules. So on the left, you see the primary diseases that cause progressive kidney disease, minimal change disease, although fortunately that often responds well to treatment and may not progress to end-stage kidney disease. It's a bit of an exception. Focal segmental glomerulosclerosis, that is a disease in which the glomeruli, the filtering units of the kidney, some are involved, hence focal, and when they're involved, there are segments that are scarred, and you'll be seeing a picture of that. That'll be a major disease that I'll be covering. Membranous nephropathy is a disease caused by antibodies to kidney proteins or antigens, responds to immunosuppressive therapy. And globally, probably the most common disease on this slide is IgA nephropathy because it's particularly prominent in Asia. So Japan, China, other Asian countries, probably due to genetic reasons, still an important disease in this country. So that's the left side of the slide. The right side of the slide are secondary diseases in which there is a systemic disease or process. So lupus nephritis, a relatively common autoimmune disease. Systemic vasculitis, or inflammation of the blood vessels, again, a systemic disease. An antibody-driven disease is anti-glomerular basement membrane disease, where an antibody is produced that attacks the glomerular basement membranes of the kidney and other tissues. And finally, various forms of proliferative glomerulonephritis, proliferating more cells enter the kidney, and I'll be touching on this. So FSGS are of particular interest in the context of African American kidney disease and APOL1. So let's dissect that term. First of all, it is focal. It initially involves just some glomeruli, and it is segmental. So you can see here at the bottom left part of the kidney, you can see scar. There's more red protein, specifically collagen, that's gumming up the normal filtering units of the kidney, the capillaries, that are still open or patent throughout the rest of the glomerulus. So this is a disease that starts with segmental scars where the rest of the glomerulus is normal. Initially, only a few glomeruli are affected, hence the term focal, and it progresses over time to affect most glomeruli. Typically, immune deposits are absent, unlike, for example, membranous nephropathy that I was talking about earlier. There may be scarce immune deposits limited to two antibodies called, or antibody classes called IgG and IgM. But this is not primarily an immune disease, although it can respond to some of the immunosuppressants that I'll be talking about, but they may not be working primarily in that fashion. Importantly, there's genetic susceptibility to FSGS as there is to a number of other kidney disease. It's been known for years that many African American families have multiple members with kidney disease, and they may be different kidney disease in different family members, including focal segmental glomerular sclerosis, that disease I showed you, that partial scarred glomerulus a minute ago, HIV-associated nephropathy that used to be rampant. Now, with effective therapies for HIV, fortunately, we're seeing much less of it. The therapies for HIV that will knock down the virus and essentially halt or prevent the disease. But not everybody is aware of the need to take the therapies or has regular access, so it does remain a problem. Also, what's sometimes called hypertensive kidney disease, chronic kidney disease, which is a very poor term, I admit, because most chronic kidney disease are hypertensive, but nonetheless, it does show up on biopsy reports, and it's important for the clinician who's reading that report to say, aha, I need to dig deeper and figure out what form of kidney disease it is, because it may be kidney disease that causes hypertension rather than hypertension causing kidney disease. Nonetheless, out-of-control hypertension, somebody with a blood pressure walking around of 200 over 140, will damage their kidney, and that is a bona fide example of hypertensive chronic kidney disease. And finally, diabetic nephropathy. I've listed it last, but it's the number one cause of chronic kidney disease in our country. So many people have diabetes. Rigorous attention to blood glucose control can usually, but not always, prevent diabetic nephropathy, and it can certainly slow progression. So again, that's a very important disease, so I won't be saying much about that today. So back to FSGS, or focal segmental glomerulosclerosis. Primary FSGS that is not caused by another condition, particularly in African descent individuals, and I'll be coming to why that's important, is best considered as a complex disease with both genetic and environmental factors. So we began a number of years ago. You can see this slide has two groups publishing in the same issue of Nature Genetics back in 2008, looking for a genetic variant that might explain why African Americans with FSGS, again, focal segmental glomerulosclerosis, compared to controls, so the controls here are in this light blue color, and the cases of FSGS are in the black lines. And we noticed a peak of African ancestry. So what this is charting across the bottom line, chromosomes 1 through 22, and then chromosome X, the presence of variants from the control genomes in cases versus controls. So let's start with the controls, the blue line. The percent of African admixture was relatively jumped around across the baseline of set at 0 for each genetic group. However, over chromosome 22, we noticed a surge of African ancestry at chromosome 22 near a gene called myosin heavy chain 9. It's not important at this point. We thought it was important at that time, but like so much of science, sometimes you draw a conclusion that proves to be wrong. But we were very interested in NIH9. Our group and also another group led by Linda Cow both published a paper in the same issue of Nature Genetics saying, aha, there's an important signal over in chromosome 22 suggesting that genetic variants in that region are a cause of FSGS. So the hypothesis was that an increased for idiopathic, that's a term that means no other clear explanation for disease, an HIV-associated collapsing glomerulopathy, a particular form of FSGS, is due to African origin genetic variants. So here you see in the center a map showing on the left side the Americas, on the right side Asia, and in the center Europe in green and Africa in yellow. And the concept was that as African Americans were brought to this continent, unfortunately as slaves, something that we all should be mindful was a terrible injustice beginning in the 1600s and lasting up until the 1800s. So the ancestral populations from Africa, Asia, and Europe and Asia differ in prevalence of FSGS risk alleles, and that we had the hypothesis that admixture mapping, that is mapping where we look at each person's genome and look for variants that came from Africa, Europe, or in some cases from Asia, particularly from Native American gene influx, can find the gene using single nucleotide polymorphisms that can tag African versus European origin genetic segments. Again, we're looking for an African origin genetic variant that could explain why today African Americans are prone to more kidney disease. I know that's a complicated topic, and I'm happy to expand in the question period. So further examination of FSGS in the kind of analysis I just showed that the strongest signal was in two variants termed G1 and G2. Notice that the first author of the first paper is Giulio Genovese, who liked the letter G. So I'm teasing a little bit here. So he named the variants G1 and G2. So this study involved from two other groups, not ours, showed that APOL1 variants were enriched in African Americans. So when we controlled for a particular SNP, we saw another signal, NYK changed called G2, and then another signal. When we controlled for G1 and G2 variants, that is, we took them out of the analysis, there was no further signal for kidney disease in African Americans, suggesting much, if not all, of the genetic risk for FSGS and for kidney disease, glomerular disease in general among African Americans was due to these two genetic variants. So what is this gene, APOL1, that carries these two variants that seem to drive much of the excess kidney disease, primary kidney disease in African Americans? As an aside, I would say I'm talking about primary disease. This does not relate to diabetic kidney disease, which is a whole different story that I won't be addressing. It's a relatively small protein. It's only about 400 amino acids. Some proteins like hemoglobin may have 1,000 amino acids or more. This gene is a component of the innate immune system. Innate meaning it's something we're born with, we have all the time, and we don't have to be exposed to foreign antigens like vaccines that were given to boost immunity, say, against polio or diphtheria. Its expression is driven by interferon, an immune response gene. It's a minor component of the good cholesterol, HDL cholesterol, so it circulates in our blood. It has a protein that regulates autophagy, it binds phospholipids, and I realize those points are really a bit beyond the scope of this talk. So the APOL1 gene family is interesting. It's evolved rapidly in primates so that APOL1, but APOL1 is the expressed gene only in humans, in gorillas, shown here as that arrow pointing to that blob, that tear-shaped blob, where you can see it's a little bit hard for me to see, but it's essentially in gorillas and humans. Very interesting. The chimpanzee is our nearest neighbor and it doesn't have it, so it must have lost it in the evolutionary process, and why this gene would have been lost from the genome is totally unknown. And it's a pseudogene in macaque, monkeys, and in orangutans, suggesting that in some contexts, the benefits of having APOL1 are less than the harms that it might be producing. Again, we don't know much about that. I'm not sure that APOL1 in humans, except for the variants I'll be talking about, but this is an interesting genetic story. Still a puzzle. So again, to develop this a little bit further, we see a variety of primates on the left. We're at the top, and we have APOL1 as well as other APOs. Chimpanzee, our nearest neighbor, has lost it. Again, a puzzle. The gorilla has it. The orangutan and macaque, that trident symbol indicates that it's been pseudogenized, so it's not expressed. So we don't really understand, as I say, this evolutionary history, but it suggests that APOL1 was an important gene for some reason for humans to retain and for gorillas to retain, but was lost in or is non-functional in other species. So some of the mysteries that still need to be worked out. So the APOL1 risk alleles, the variants, protein variants that cause kidney disease, as I mentioned, are termed G1 and G2. And this is a map, obviously, of the world. You see these pie-shaped signals. On the left, over America, you can see the fraction of African Americans that carry the G1 allele, a risk allele for kidney disease, in red, and the G2 allele, also a risk allele, in tan, although most African Americans carry no copies. And European Americans and others, as you can see going down into Africa, do not carry any risk alleles. Now, these are self-identified European Americans and African Americans. And these days, I would argue it's important for anybody with kidney disease in America to be tested for APOL1, because we don't always know who our grandparents and great-grandparents and great-great-grandparents are, and I don't think we can fully exclude the possibility that some of us have undetermined African ancestry. So in this day of whole genome scanning, this is an important gene to be looking at in somebody with kidney disease in case they might be a rare individual who doesn't know about their African ancestry. Moving now to Africa, you can see a band, sub-Saharan Africa, where various groups in the population have G1 or G2, the two risk alleles, and it in general is absent from Asian populations. So again, it's an African origin genetic variant. So why do we carry these variants? Well, it's to protect us against infectious disease. If we go back 10,000 years ago, the things that tended to kill us were trauma and infectious disease. We didn't, in general, live long enough to develop cancer, I believe. Don't know that for a fact, but I think that's quite likely. And we didn't eat diets, in general, that were high for heart disease. And so many of us probably died relatively early of various things, including trauma and infectious disease. So trypanosomes, as an example, would be an important cause of disease that we would be prone to. So on the left, you see a mosquito, which carries a form of trypanosome infection. And this cow, I'll say, I don't know for a fact, but we can posit that perhaps it has chronic disease that's causing it to be wasted. The other two, TB gambiense and TB rhodesiense, again, trypanosome brucei is what those letters stand for, circulate in our blood if we're infected. So you can see that blood smear with the red blood cells showing up as these circular cells. And you can see a semicircular, if you will, protozoan that's in our bloodstream. Slightly different forms in the West Africa and in East Africa, or Southern Africa, shown with those names. And we see a child here sleeping under a tent, so that would be a mosquito tent, that would nonetheless potentially have been infected by these trypanosomes. So APOL1 is the trypanolytic factor of human serum. It's our defense against the trypanosome that enters our bloodstream. It rides on HDL cholesterol particles, shown in the left, where the HDL particle has an APOL1 protein, that blue blob, and when it's taken up by the organism, the trypanosome, which is the major structure we're seeing, it kills the trypanosome. Unfortunately, the trypanosome involved a countermeasure in the ongoing struggle between host and parasite to get on top. And so the trypanosome involved the protein serum resistance antigen that prevents APOL1 from killing. We, in turn, evolved variants in APOL1, and my next slide may be covering this as well, variants in APOL1 that evaded that defense, but unfortunately, those variants in APOL1 cause chronic kidney disease. That's a major theme of this talk today. So again, we all have APOL1. The trypanosome involved a countermeasure, serum resistance antigen. Our move on the evolutionary tennis game, if you will, would be to evolve variants in APOL1 that restore killing. Good, we don't have trypanosomal infection of that sort, but bad, it causes chronic kidney disease. So there's an alternate view. How exactly does the trypanosome get killed? And there's a view that it's via a cation channel. For our purposes, I think that's a detail we don't need to go into. So to summarize, we all carry APOL1. APOL1 variants developed the serum resistance antigen was developed by a form of sleeping sickness organism, TB rhodesiense. And in turn, we were selected, people in sub-Saharan Africa were selected to carry variants in APOL1 called G1 or G2 that restore killing, so that rhodesiense organism is kind of looking sickly because it's been killed. And it's a little less clear if it's effective against gambiense, which is why that question mark is there. So the take-home message of this somewhat complicated story is that we evolved variants in APOL1 that were able to kill otherwise resistant parasites, but those important variants called G1 or G2 unfortunately cause chronic kidney disease. So what's the distribution of APOL1 risk variants? Very complicated slide, but if you'll focus perhaps on the text on the left, G1 allele is highly frequent in West Africa, as shown by the blue portions of the circles. You can see it around West Africa, which is where the majority of African American ancestors would have come from. The risk allele frequency, the APOL1 risk allele variants, G1 and G2, are 55% in some of the peoples of West Africa, the Yoruba, Igbo, and Asante peoples. And as a result, and this is important, about 35% in African Americans, 35% of an African American population will have risk alleles. About 13% will have two risk alleles, which is the status that is most likely to cause kidney disease. By two risk alleles, I mean G1 and G1, or G1 and G2, or G2 and G2. Again, we all have two chromosomes, 22, where this gene is, and about 30% of African Americans have two risk alleles, and that's the most common genotype to drive kidney disease. It is true in certain situations, a single risk allele, APOL1 risk allele, can cause kidney disease. But this is the 13% figure. I think I have worked out the math later. It's several million people in our country and millions more in sub-Saharan Africa, in South America and Central America, carry two risk alleles, APOL1 risk alleles, and are much more prone to get kidney disease. What else have I said here? The G2 allele is older, more widespread, and it has additional protection against brucei, but allows more severe disease with gambiense. Again, the trypanosome variants. For our purposes, that admittedly is a somewhat subtle point. Okay, so this slide illustrates the point that APOL1 is under balancing selection. It's best to have just one copy. On the left, you see individuals who have no risk alleles. They have so-called G0, G0. This is the homozygous disadvantage. This would be true of populations solely from Europe or Asia. They are prone, if they are in an environment where they are exposed to APOL1, they are prone to get African sleeping sickness, but they don't get kidney disease from the gene itself. On the right, you see individuals with two risk alleles, G1, G1 or G1, G2, who has homozygous for these genetic variants. They are more prone to get chronic kidney disease, a major theme of this talk, but they are effective against trypanosomes. The middle group is sort of the sweet spot. This individual has a single copy of the variants G1 and G2 that fight the trypanosome, but they can, in some cases, get kidney disease. But it's a heterozygous advantage because the kidney disease is much less common and they're protected against trypanosomes even if they live in sub-Saharan Africa. So what are the APOL1 nephropathies that is seen generally in people or at risk for people who have two APOL1 copies? Well, the highest prevalence is in people with HIV-associated nephropathy or HIVAN. The risk in individuals with 2 risk copies is fully 29 in the United States and 89 in South Africa. So again, carriage of 2 copies of the risk variants poses an enormous increase, as you can see, particularly in South Africa, to develop chronic kidney disease. A much more common disease, particularly now that HIV is coming under control, is focal segmental glomerulosclerosis. Still, a person, generally African American, with 2 risk copies has an odds ratio in their lifetime of 17 to develop chronic kidney disease. Somewhat less hazardous, with an odds ratio of 7, is what I'm calling here arterionephrosclerosis, sometimes called hypertensive kidney disease. But in these individuals, it's not driven by hypertension. They may have hypertension as a result of their kidney disease, but it's driven by these risk variants. And unfortunately, it often progresses to end-stage kidney disease. The next one down is, what about kidney transplant? The deceased donor genotype can impact the outcomes of the recipient, so that the allograft loss in an individual who has APOL1 variants in the donor in the recipient can have a hazard ratio of 3.8 to lose that kidney. So it's not an ideal kidney to receive as a recipient, although it may be better than waiting much longer on dialysis to get a kidney that's free of any APOL1 variants. Importantly, for the recipient of an APOL1 risk allele positive kidney, there's no effect, because the risk is unaffected by the recipient genotype. All of the effect of the APOL1 variants travel with the kidney. How about sickle cell? An individual who has sickle cell often is an African American, and they may also carry APOL1 variants. If they carry 2 variants, the relative risk is 3.4. And it is important to consider in a patient with sickle cell disease that their kidney disease might not be just due to their sickle cell, but potentially APOL1 might play a role. And lastly, eclampsia, a severe form of kidney disease that begins as preeclampsia, but full-blown eclampsia, the relative risk is 1.6 in individuals, odds ratio of 0.9, in individuals, generally African American, who have risk alleles for APOL1. So that's an important thing to test for in an African American mother who develops preeclampsia or eclampsia. So we carried out a study in African Americans to look at the prevalence of FSGS associated with APOL1. So on the left, we see the FSGS control group, where 13% of African Americans carry 2 APOL1 risk alleles. If we looked at the bottom with so these were control individuals who did not have kidney disease. That's the general population frequency of APOL1 high-risk status in African Americans. In FSGS cases, African Americans who had progressed to develop kidney disease, fully 72% have 2 APOL1 risk alleles. On the right, we look at the setting of HIV-associated nephropathy, also in African Americans. And in those who, by control, I mean did not have kidney disease, about 8%, very similar to that 13% figure, had 2 APOL1 risk copies. In the bottom right, you look at HIV-associated nephropathy, where much like those in the FSGS cases, about 72% of African Americans had 2 risk alleles. The risk allele frequency in the population is about 36%. Squared, that comes down to about 13%. So you can see the take-home message is that in those with FSGS or HIV-associated kidney disease, there's a much greater prevalence of APOL1 high-risk status. And so as we treat patients, we need to think about, with FSGS or with HIV-associated nephropathy, whether we need to be thinking about APOL1 risk alleles and whether therapies against APOL1 might contribute to their treatment. And I'll be coming to that in a minute. What do we know about when APOL1 disease occurs in the life course? And the take-home message is shown in the top line. There's a sharper incidence peak of onset of APOL1 disease in the ages of 15 to 45, sort of the prime of life. So let's look at the panel on the left. Let's look at patients with FSGS, this is the blue bars, and say, when does non-APOL1 FSGS appear, and the peak appears to be about 40 to 50. If we look jumping over to the right, we see a fairly similar pattern for European American FSGS. And then in the center, we see those with two APOL1 risk alleles. Overwhelmingly, these would be with known African ancestry. And you can see there's a sharper disease peak, incidence peak, roughly between the ages of 20 and 40. So sort of the prime of life is when APOL1 associated FSGS tends to appear, but it can appear at most ages, as you can see from this. So there's an association with APOL1 risk variants with HIV kidney disease and with FSGS. So this sort of summarizes a lot of data. If we look at HIV-associated nephropathy in the United States, 72% of individuals have two risk alleles, and there's an odds ratio of 29. If we look in a second study, very similar, 62%. If we look in South Africa, a similar number, 79%. So again, in the setting of HIV, a very high prevalence of kidney disease, and the message here, or the mechanism here, is likely that interferon, which this virus, HIVAN, causes the body to produce, also drives expression of HIV. By contrast, if we look at another HIV disease, HIV immune complex disease, there's not much of a signal for a role for APOL1. That's true in both U.S. cohorts of HIV patients and South African cohorts. If we look at HIV-positive FSGS, which is a slightly different variant, less collapsing features on the biopsy, we're back up to 63%. So a large, high prevalence of APOL1 2 risk allele status among individuals who have HIV-positive FSGS. And in HIV-negative FSGS, so this would be patients who have HIV is not in the picture. They may have a variety of diseases. They may have been called hypertensive kidney disease. These individuals have a very high rate of 2 risk allele status. So once you know your patient, if you knew genetically what their patient genotype was, you could tell them your risk to progress is much greater, shown here in the red line, so that those go to end-stage kidney disease with a GFR of 0 or close to 0 within about 15 years, whereas individuals who have 0 or 1 risk allele tend to have a much slower rate of progression. Both are progressing to kidney disease, so everybody who has FSGS should be taking the therapies that are appropriate for them for FSGS, and if they have APOL1, for APOL1 therapies. But particularly somebody with APOL1 needs to know they don't have a lot of time to address this problem in addition to treating their FSGS, so specific therapies that target APOL1, because end-stage kidney disease may come quite quickly in this condition. So how about patients with FSGS, focal segmental glomerulosclerosis? How do they respond to the therapies that we commonly use for focal segmental glomerulosclerosis? So if we look at prednisone, an immunosuppressant, it is similar in those who have the high risk allele status compared to others. Another immunosuppressant drug, cyclosporin, similar rates of response between people who have 2 risk alleles and others. How about mycophenolate, another immunosuppressant? Again, similar, and the names of the trials are put there in parentheses. We don't know about rituximab, a commonly used antibody. I suspect it's the same, but that's a study that I haven't seen being done. So the message is, if your patient has a high-risk APOL1 genotype, they still need to receive the same therapies that other patients do, with the caveat that these are immunosuppressants, and so if they have other complications that they need to be thinking about closely, but in general they would receive the same therapies that patients with low-risk genotypes would. Importantly, APOL1 risk alleles with high risk tend to progress quickly, so we can see faster progression, the red line going down across the various slides. So the message here is that a patient who comes to you with FSGS, high risk allele status, that patient needs to have the understanding that they may progress quickly to end-stage kidney disease. There may not be a lot of time to bring it under control, and so they should take their medication. If they have side effects, they should let you know as a physician, and be able to move on to another medication. And the patient has to understand the importance of effective treatment and trying to get it under control. Okay, the African American study of kidney disease and hypertension was done nearly over 20 years ago now. It studied about 1,000 African Americans. It randomized them to two groups, to more effective blood pressure control and to tighter control versus less control, and two drugs, amlodipine versus metoprolol, actually a third drug, ramipril. 23% had two APOL1 risk alleles, which is the highest group status. And then the main message here is that the high-risk status was associated with more proteinuria and a higher creatinine, meaning they had more advanced kidney disease when they were enrolled. So again, a signal that this is a disease that tends to progress relatively quickly. It's important to make the diagnosis early and to employ some of the new therapies we'll be talking about quickly to try to bring the APOL1 component of their disease under control. So in this study, progression in the African American study of kidney disease occurred even with intensive control in APOL1 high-risk patients. So it's a somewhat complicated slide, but the two black lines show the rate of progression, loss of glomerular filtration rate, in those who are low risk status 0 or 1 risk allele or lower. Those who have 2 risk tend to progress more quickly, so those two red lines are dropping fast, even in those who are at the intensive treatment goal. So again, the message is hypertensive patients need to have their blood pressure well controlled, particularly if they have progressive kidney disease, but those who have APOL1 positive disease also need additional therapy that we'll be coming to, because you can see them progressing despite having intensive therapy. So the message from the last slide is normalizing blood pressure did not stop CKD, chronic kidney disease progression, in the African American study of kidney disease and hypertension. That led many of us to be rethinking hypertensive kidney disease, that it's more than just hypertension. There's a role for arterionephrosclerosis, which the hypertension may be driving. There may be genetic component, in this case APOL1, and there may be a metabolic and inflammatory disorder that causes the hypertension. So progressive hypertensive so-called kidney disease is complex, and we need to be addressing metabolic factors, type 2 diabetes if the patient has it, metabolic syndrome, obesity, lipids, elevated uric acid, certainly inflammatory causes, smoking is key, diabetes is important, HIV, and aging. And genetic causes, APOL1 contributes. So it's a mixture of causes that drive progressive vascular disease, that drive so-called hypertensive kidney disease. So in fact, the hypertension may be downstream of many of these input factors that are causing damage. And then the outcome is a failure of the normal regulatory processes in the arterioles, leading to ischemia, activation of the renin-angiotensin system that then contributes to hypertension. So a complex set of input factors that are driving damage to the kidney. It also means a complex set of factors that potentially we can address as we dissect in a given patient what may be most important. APOL1 variants can cause FSGS, and these can manifest as 4 different mechanistic forms of FSGS. In the first, primary FSGS, common particularly in children and young adults, can be responsive to steroids and can recur after kidney transplant. It is, after all, a genetic disease. Why would it recur after kidney transplant if it's a genetic disease? That's a little bit of a puzzle. Certainly the donor kidney needs to be thought about. But as I say, the recurrence is still a puzzle. Maladaptive glomerular processes, for example, somebody with sickle cell disease can drive kidney disease. CKD associated with prematurity. HIV-associated nephropathy we've talked about. And interferon therapy, where the link is the clearest, where interferon drives APOL1 expression in the kidney. Implications of the observation that different factors can contribute to APOL1 kidney disease. I'd offer what might be called the fertile soil hypothesis, that podocytes and perhaps vascular cells are susceptible to injury with APOL1 variants that alter endolysosomal function or inflammatory signaling or some other process. So, for example, in primary FSGS, believed to be due to a cytokine, that may be driving it, and if there's interferon is upregulated, then certainly that potentially could bring in APOL1. HIV drives interferon expression and also may affect podocytes, may infect podocytes. Arterionephrosclerosis, there are contributors from blood pressure and lipids and inflammatory factors. And lupus is an interferon-positive disease, and that could bring in APOL1. And in type 2 diabetes, glucose, blood pressure, and lipids would all be compounding factors potentially in an individual who has APOL1 risk factor variants. So the Neptune study, the nephrotic disease study that I mentioned before, has the advantage that renal biopsies are done on a research basis at the time of diagnosis of a glomerular disease. And this allows us to understand early in the disease process more about how APOL1 risk alleles are changing gene transcription, that is, RNAs being produced that then make proteins that can alter the biology. So comparing high risk, that is, two people with two APOL1 risk alleles, and low risk subjects with 0 particularly or 1, the three most upregulated genes were small cytokines, CLCL1 and CXCL2, and both of these are interferon regulated, and another protein that I know much less about, UBD. Among the activated pathways that may be downstream from some of these small proteins are JAK family kinases involved in cytokine signaling, STAT signaling, and ILK signaling. Now, I know this is complex, but it begins to be a window into how APOL1 is operating on the genome to increase potential expression of proteins that may drive kidney disease. And if we can't successfully bring down APOL1 expression, potentially some of these intermediate targets could be choices that we might choose to attack. But clearly, addressing APOL1 variants would be the most direct way to address the effect of APOL1 variants. So how about donor genotypes? This is an important issue, and this particularly looks at APOL1 variants in deceased donors and how they fare. So this was 55 U.S. medical centers, over 600 kidneys, and the risk of an individual kidney from a high-risk individual who has died and generously provided their kidney is about 2.3 for kidney loss, which is highly significant. Much more significant than simply looking at somebody who's African American who has died, where the hazard ratio is only 1.6. Still significant. So the take-home message is, and you can see on the left, the survival of those kidneys is worse with those who have two risk alleles. It's not actually much worse, and if you had to choose waiting another few years to get a kidney or getting a kidney from a high-risk donor, you probably might want to go with the high-risk donor, but if you had a choice, you might want to go with the low-risk donor if you didn't mind waiting. So it does play a role, but compared to everything else, it's a much less of a role to receive a kidney from a high-risk APOL1 risk allele donor. So how about the recipient genotype? So you're an individual who unfortunately had two APOL1 risk alleles. You progress to end-stage kidney disease. And does it matter what your recipient genotype was? And the answer is it doesn't. You do just fine with the new kidney as somebody who doesn't have risk alleles. So the key here is that the risk for recurrent disease travels with your original kidney and not with your new kidney. And so you shouldn't be hesitant as a transplant recipient about proceeding with transplant. Ideally, you would get a kidney from somebody who didn't have two risk alleles, but your past history of having two risk alleles in your own kidneys has no effect on the outcomes of the new transplant. How about living donor outcomes? So this is so important. We're taking healthy people and asking them to donate their kidney to a child, a spouse, a sibling, or maybe just another person. So this looked at 136 African American living donors. High-risk allele was seen in 19 and low-risk allele in 17. This is a small study. These were African Americans, so you can see the rate of carriage of these donors with high-risk allele was relatively high. The median follow-up was 12 years. African Americans, high-risk donors, had lower GFR before donation because there was some impact, even though they were screened for kidney disease, there was some impact for modest, for less kidney. And after donation, there was still a difference, but modest. But importantly, two donors developed end-stage kidney disease. Both were high risk. So you see that on the risk on the right side, you see the GFR before donation was pretty similar, but there was still a significant difference with the low risk having slightly higher GFR, 0.04, and that persisted after donation. So what's the take-home message? An individual who is considering being a donor and is African American and is tested for APOL1 variants should think carefully about whether they want to proceed. They might say, I'll take that risk of a somewhat lower GFR. It's my child, it's my spouse. But they might be less motivated if it's a random individual that they might donate to, and they might say, I don't want to put myself at any risk to have a lower GFR at a number of years after donation, although they might still go ahead. It's just that they need to be counseled and to come to an informed decision about whether or not to proceed. So summary of key clinical features as I summarize here of APOL1 kidney disease. It can manifest histologically as focal segmental glomerulosclerosis, collapsing glomerulopathy, a form of that disease, and arterionephrosclerosis. It can present as primary FSGS, heavy proteinuria, low serum albumin, and maladaptive FSGS, where the scarring has occurred, but they may still have relatively normal levels of creatinine and protein for a while. Collapsing FSGS variant is particularly associated with APOL1 variants. Interferon is a driver of APOL1 gene expression and therefore explains why certain viral infections may be associated with more APOL1-associated disease. The disease is more advanced at presentation and has faster progression than other forms of FSGS, so there's a shorter window of opportunity for intervention. So important for the clinician to recognize by genetic testing that their patient has APOL1-associated FSGS or collapsing glomerulopathy or arterionephrosclerosis so that they can be afforded treatment with therapy that targets APOL1. Response to steroids such as prednisone, cyclosporin, and mycophenolate interestingly appears to be similar to other forms of FSGS, perhaps because these are immunosuppressants that can tamp down APOL1 expression. And kidney transplant donor, 2-risk allele genotype, is associated with worse allograft outcome and again may also put the donor at some risk. Approximate risks for APOL1 nephropathy, there are about 43 million African Americans. About 13% or 5 million have 2 APOL1 risk alleles. Among those, perhaps 20 or 25%, and this is just an estimate and may not be exactly correct by any means, but approximately 20% will develop a kidney disease or kidney phenotype associated with APOL1 variants. So perhaps 5 million individuals are at estimated lifetime risk that is increased. So what are those relative populations that are going to get at risk? So FSGS, perhaps a 4% lifetime risk. Hypertension attributed end-stage kidney disease without a biopsy that might have been FSGS that wasn't diagnosed. Or hypertensive attributed CKD. Again, it may not necessarily be all hypertension. And type 2 diabetics who also have APOL1 risk alleles. So the message is for the clinician, there are various subgroups who will manifest different ways with APOL1 high-risk status, and these need to be thought about by the clinician. So how about incorporating genetic testing into precision medicine? The latter term meaning we have a more precise conception of what is driving the disease in terms of genetics, social risk factors, drug exposures, a host of things that help us understand why a particular person got a particular disease. When we have effective therapies, well, we now have those. Implement screening for APOL1 risk status in all CKD, I would argue, because we don't always know the genetic background of our patients. Education for physicians, particularly primary care physicians and internists who may be the first to see these patients before they get to a nephrologist. And one of the interventions is inaxaplin, a Vertex drug, but must be widely available. The cost is now about $400 a dose, and I think insurance companies would pay for it, but not everybody has insurance. In other countries, this may be a price that's out of reach. So recent developments, I've mentioned inaxaplin, adalimumab, sparsentan. I think I'll say a few words about some of these. So I did talk about inaxaplin, I believe. I duplicated this in this version of the talk. But basically, it's an APOL1 antagonist. It's in phase 3 trials at the present time. You can see there was a New England Journal paper. The FDA has given it breakthrough therapy status, which could speed the eventual approval process. It will still be rigorous, but it may not require quite as many patients or quite as many trials that show efficacy. And drug cost was not available that I could find it. I'm sure it will not be inexpensive, as this is not necessarily going to be a very large population. Another drug, adalimumab, is a monoclonal antibody that targets tumor necrosis factor, TNF-alpha. There are 3 phase 1 trials, in other words, often single-arm study, sometimes with rising dose. All trials supported safety. Next step would be, and the most recent one was 2023, so the next step would be a randomized controlled trial, and I don't have any information about when that might potentially be started. But encouraging that we have several drugs on the horizon. Sparsentan is a small molecule from Travere Therapeutics. It blocks receptors for endothelin A and also for angiotensin type 2, which would relate to the effects of angiotensin on the kidney. There are 2 studies, Duet Phase 2, Duplex study completed in 2023, well tolerated. There's not a lot of published data yet, particularly from the Duplex study, so we'll see whether this is in fact a promising drug for APOL1 disease. So APOL1, look ahead. Increased awareness of APOL1. We need to have that. We need APOL1-associated disease in the African descent communities, health fairs, churches, employers, so people understand that an African American with hypertension, maybe a little bit of proteinuria, may not just have uncontrolled hypertension, may have APOL1 kidney disease. And so I believe it's better to spread the net widely and try to identify these individuals. And once you find an individual who's at a high-risk status, their relatives also need to be tested. So this is an enormous effort, but I think it's one that we have to make as a society to find out who is at high risk, get them the medications, and we have medications now, and I'm sure we'll have more in the future. So there's hope, but a lot of it begins with screening to find out who out there could benefit. So education of healthcare providers. I don't think every primary care physician in the country is aware of this, and they can't be. They need help getting this news out. So I think people in the community can be helpful in terms of having posters at health fairs, finding ways to have speakers at physician conferences to get this in the forefront of people's minds, both clinicians and the general society, that this is something we should be looking at. Just like we screen for diabetes, screen for hypertension, we need to be screening more for proteinuria and not just assume that it's something that is not important. Cascade screening refers to when an individual with APOL1 high-risk status is identified, offer genetic testing to relatives, because blood relatives are almost certainly going to, in some cases, have the genetic variants as well. So conclusions, FSGS comprise various syndromes. I've talked about some of them. It involves diverse initiating causes, mechanisms, clinical courses, treatments, and outcomes. It's not one disease. We need, as a clinician and as patients, to figure out exactly what is operative in a given patient. It offers opportunities for creative investigators to make new discoveries and move the field forward. We've made a lot of advances in the last 10 years, and I certainly hope we continue to do that. So with that, I'll just acknowledge my group at NIH, my close genetic collaborator for many years, Cheryl Winkler, and her husband, George Nelson, and then some people from my lab, which, as I say, I've now left NIH, but these are great individuals to work with and certainly helped me tremendously in getting this work done, and were a pleasure to work with. Thank you to AAKP and George Washington University for the opportunity to talk to you today.
Thank you, Dr. Kopp, for your excellent presentation. Before we close, I have few questions. When AAKP initiated the Decade of the Kidney in 2019, a focus for 2020 to 2023 efforts was early identification and diagnosis of kidney disease that include rare and genetic conditions. How far do you feel we've come in early screening and diagnosis in the last 5 years? And where do you see opportunities for more over the next 5 years?
That's an excellent set of questions, and I think we've made considerable advancement. I think we're doing more screening for early kidney disease, looking at potentially unaffected family members to identify cases before they even become symptomatic. And I think we've also made progress in identifying novel therapies that we can use for these diseases above and beyond what we've been using recently.
How are researchers making sure that people most affected by APOL1 are included in clinical trial and research studies?
That's an excellent question. I think the kidney research community is very much focused on this disease APOL1 because it's so prevalent in an underserved population and because we have an effective therapy now. So I think there's great interest in screening for APOL1 variants and identifying patients early and getting them started on therapy. So I think we will see even more progress in this direction in the years ahead.
What role can patient and care partners play in advancing APOL1 research and awareness in their communities?
So I think patients and care partners, and in fact others who may not be neither individuals, but are aware of this disease, can play a great role in spreading the word in their communities, churches, synagogues, other social places where people interact socially, to let people who have not heard about this story become aware of this and reach out to their clinicians to learn more about APOL1 disease. And if they are at high risk, be screened regularly. And if they have proteinuria or signs of kidney disease, to begin therapy with a range of therapies, but particularly targeting APOL1.
Thank you again, Dr. Kopp, for your insight remarks. We greatly appreciate your sharing your time and expertise with us to discuss the important research you are conducting in APOL1.